What I want to do this morning is to walk you through some of the things that we're learning about the structure of Cas9 and how we think it actually interacts with DNA in a structural sense. And then I want to tell you a little bit about how we can use that structural information to think about engineering the protein to do different things. And then I'm going to talk about, on request, about work that is ongoing to engineer Cas9 to recognize nucleic acids that it doesn't normally interact with in nature, namely RNA molecules. So this is an outline of what I want to talk about. And so I'll dive right into thinking about the mechanism of surveillance. And this kind of builds on what we were talking about yesterday. So I showed you this slide yesterday, and I just wanted to revisit this for a moment. So currently, I would say that the data that we have both in vitro and coming from some of the live cell imaging experiments that I talked about in yesterday's lecture suggest that the way that this protein RNA complex interacts with DNA is not through a sliding kind of binding and sliding kind of mechanism, a processive mechanism, but instead we think that this is actually somehow a complex that's able to bind and release DNA very rapidly. And if there is processive interaction with DNA, we think that processive interaction would have to be along very short segments of DNA. We can't rule that out. It could be short sliding events that occur, but we don't think it's a protein that gets on and then slides from one end to the other of, say, a chromosome. So there's very rapid binding and release, and as I mentioned yesterday, there's a binding site in the protein for this motif called the PAM, which for this particular enzyme is a GG dinucleotide. And our data would suggest that when this protein interacts with a GG dinucleotide, that interaction at least transiently reduces the mobility of the release of the rate of release of the DNA from the complex. And that interaction then might provide time for initial base pairing interaction between the guide part of the RNA and the DNA. And I'm showing it here as we sort of imagine this to be involving a local unwinding of the DNA. And I don't really understand the details yet of how that works, but somehow there has to be invasion of this RNA strand, and that unwinding of the DNA, I think, is coupled to the base pairing with the RNA, such that if there is complementarity to this guide strand, eventually this kind of structure forms, an R-loop type structure that puts the active sites of the enzyme in a position to generate a double-stranded break in the DNA. And it's really this sort of model is guiding our thinking, certainly, about how to design experiments to really test this kind of mechanism. Is this really what's going on? And if it is, if it's not, we'd like to revise it. And if it is, then we'd like to understand the details better for this. And so I want to show you this is another sort of cartoon type slide that summarizes some published work that represents a series of structural experiments that were done to investigate the way that this protein, sort of the conformation of this protein as it interacts with nucleic acids and assembles ultimately with a substrate DNA molecule. And this was using a method called negative stain electron microscopy. And what you do in that approach is you take a molecular sample and we can array it on or basically apply it to grids that can be imaged using electron microscopy. And in this kind of experiment, we're using a stain that will basically coat these molecules and allow us to detect the structure of the molecule. It provides nice contrast to the underlying solution that the sample is embedded within. But the stain also means that the resolution of that kind of experiment is not very high. So this is really a cartoon because our actual images are sort of about like this, sort of very blobby images, about 30 angstrom resolution or so. And then what was quite remarkable in this set of experiments was that even at that low resolution, we could see a very pronounced change in the structure of the Cas9 protein in going from the protein alone over here to conformation that it assumes when it's bound to the guide RNA and then ultimately with DNA. And so what I'm showing here is, so the experiments that were done, if you're interested, you can read about it here. These experiments were done by putting different kinds of labels on the parts of the protein so we could figure out where we kind of orient ourselves with respect to the catalytic domains of the protein. And so in this cartoon, we found that the catalytic domains were localized on one side of the protein, we call it one lobe of the protein in blue, and then there was a larger separate domain or lobe over here in gray that was involved in interaction with the RNA when the RNA binds. And it's these two parts of the protein that rearrange to form a channel down the center of the protein that is the binding site for the RNA-DNA hybrid that forms when the DNA substrate molecule assembles with the complex. And so this was kind of the first indication that there might be something very interesting going on in terms of the structural dynamics of this protein and how it interacts with DNA. And one of the things I wanted to just point out, I didn't say this explicitly yesterday, but one of the fascinating things about Cas9 is that it's a helicase, right? It's a protein that's able to unwind DNA locally, but it doesn't hydrolyze ATP in the process. So most enzymes that have to manipulate DNA, whether they're helicases, other kinds of helicases, or repair enzymes, or enzymes that move along DNA in catalyzing replication or repair, they often have to hydrolyze ATP to provide the energy to do that. And here we have a protein that can unwind 20 or more base pairs of DNA without hydrolyzing ATP. So what provides the energy for doing that? And I want to suggest to you that really there's something special about the conformational rearrangement of this protein that is part of the answer to that question. So I'm going to show you two movies that now take that very low resolution model that we had of the conformational rearrangement of Cas9 and now use crystal structures that are available. There's now several crystal structures of Cas9 in different states of nucleic acid binding that we can then we can use to ask, how does the structure change in going from protein alone to RNA-bound and DNA-bound forms. And so I'm going to show you first a movie that morphs between two different structural states of this protein. First is, starting off here, is the protein alone. So this is the Streptococcus pyogenes Cas9 protein without any nucleic acid bound to it. And then we'll morph to the structure bound to a guide RNA and a single-stranded DNA target, okay? And you'll see that there's a very interesting change in the structure. So this is the protein alone in this image, and the catalytic parts of the protein are down here. Here's this larger domain that's responsible for interacting with a lot of the parts of the guide RNA. And then as we morph to the structure bound to RNA and DNA, you can see this very large rearrangement of a structural domain in Cas9 that creates the channel where the RNA-DNA hybrid ends up. And so the RNA is sort of snakes across the protein. It forms a very high affinity interaction with the protein. And this will go back and forth one more time. So you can see this very large hinging motion that rearranges the structure of Cas9, especially in this domain over here, to create the binding site for the RNA-DNA hybrid. And so this, sort of at least, it's a movie that is, morphing between these two different structures, but it certainly suggests that this protein undergoes a large dynamic change in conformation when it interacts with nucleic acid to form what we think is a very high affinity interaction with DNA. And as I mentioned yesterday, we think that even after cleaving the DNA, that this structure, we think, is maintained and has a very high affinity for the product of the cleavage reaction. something else that would have to kick it off. Yeah, question, yeah.
Have they looked at local energy minimums of the two states when it's closed and open? Is it like a pre-stress structure that allows it to, that's the energy that's provided for that?
Yeah, that's a great question. So the energetics of these different states of the protein, not in the kind of detail that would probably be nice to do. I mean, in data that I won't have time to show you today, we've been doing experiments where we put pairs of dyes, fret dyes, on the protein at different sites where we can monitor this change. And that was done initially just to test, is this really true, or is this a crystallographic, have we trapped something in a crystal lattice? You always have to, think about that. And in fact, we see evidence directly, by monitoring these changes in fluorescence resonance energy transfer between dye pairs, that there is a big change in conformation. But we haven't actually gone back and measured. I'm not quite sure. How would you do that? Would you do that by calorimetry or?
I would just think initially by computational.
Yeah.
Just to see if anything is
Yeah.
possible.
Yeah. No, I think that could be very interesting to do, because it really does suggest. I mean, I've sort of been thinking about this almost like a spring-loaded protein, right? It's sort of there's a.
There's a lot of pre-stress structure
Yeah.
in nature that provides energy.
Right.
Maybe it is the RNA finding too.
Yeah. I mean, I think our data would argue that the biggest change in conformation happens when it binds the RNA, but then there's a further change upon interaction with DNA. So. So let me show you. I'm going to show you another movie that similar kind of morphing movie that shows you in a little more detail what this hinging motion really looks like in the protein. And so this starts off again with the protein in its pre-bound state. So just the protein alone. The catalytic domains are here. And you'll see the motion that this protein undergoes when it binds to both the RNA and DNA is kind of this hinging kind of motion. You can see sort of around the base of this long alpha helix that runs through sort of connects the two halves of the protein. This region right here is really the hinge. And you can see that there's also movement of these other domains. There's a domain right here that undergoes a very large change. You saw this on the last morphing movie. And that domain ends up capping off the RNA-DNA hybrid that forms in the fully loaded structure. So I think it's very suggestive that this is somehow involved in, in the helping the RNA guide strand to invade the DNA duplex, and we'd love to understand better the energetics of that, and really what triggers unwinding. I think one of the reasons to be interested in this, of course, is in thinking about off-target binding of Cas9 or cutting of DNA at off-target sites. We'd love to understand really what defines a target site for this protein, and how can we ensure that it engages only on sites that are that are fully on target sites. Okay, so this is back to I showed you this cartoon, and so currently, right now, there are two fairly high-resolution crystal structures of two different Cas9 proteins in this state, and these are both in this paper here. One is from Strep pyogenes. The other Cas9 that was crystallized is from an organism called Actinomyces. It actually causes dental caries, and for some, Harvard Dental School had actually done a lot of sequencing of these types of bacteria. Some of them have CRISPR loci, some don't. And the ones that have a CRISPR locus actually have a variant form of Cas9 that's quite a bit smaller. It's about 25% smaller than Streptococcus pyogenes Cas9. So it was sort of interesting to have a look at the structure of that protein. And but those proteins are both in this state over here. So it's sort of this pre-bound state. And then there also are now two crystal structures of Cas9 bound to DNA. They're actually not the structure that's illustrated in this cartoon. They're actually both of them are structures where the protein has a guide RNA and a single strand of DNA bound to the guide, so we can see the RNA-DNA hybrid. And in one case, there's actually a little part of the DNA over here that includes the PAM nucleotides. And that was very nice because you could actually see the binding site for the PAM nucleotides. And then what's been missing up until recently was the structure of the real surveillance complex of the Cas9 protein bound to a guide RNA. And I wanted to show you that. So that structure for the strep pyogenes protein was solved recently by someone in my lab, Fugo Zhang, and this has actually now been published. And I want to just show you a movie that illustrates a couple of the features of this that I think are interesting and relevant to our discussion here. So first of all, this in this crystal structure, so the protein, you can sort of see this clamshell type shape, and here's the one end of the guide RNA protruding from the protein. And the actual targeting sequence in the RNA is right here in this tan color. One thing that was very interesting in this crystal structure was that even though there's a full 20 nucleotide target recognition sequence in the guide RNA, we can only see 10 of those 20 nucleotides. And the 10 that are visible are the 10 that are closest to where the PAM interaction occurs. And that's the binding site for the PAM is sort of over in this region of the protein. So I'm going to show you this movie. And so first of all, zooming in on the guiding part of the targeting part of the RNA, you can see these sort of the alignment of the bases. There's the actual electron density for that part of the RNA. After this point, it's just gone, right? So it's very mobile in the structure. And then we'll look at this in a space-filling model, so you can see the cleft that this guide part of the RNA is sitting in. And what's interesting is that the first couple of nucleotides of the guide are exposed to solvent, so they're available for interaction with a target DNA, presumably. Now I'm superimposing it on the structure solved by Martin Ginek's lab with bound to a DNA target strand. So the target strand of the DNA is here. And in his crystal structure, he included a smaller piece of DNA right here that includes the PAM nucleotides. And so this was nice because you could see the interaction between the protein and these PAM nucleotides. And I'm not showing it to you explicitly here, but it's actually 2 arginine residues in particular that come in and interact with those Gs in the DNA. And so in this surveillance structure, those PAM recognizing residues are sitting in the same, they're basically in the same orientation that they end up in the DNA bound complex. So it looks like they're really poised for this interaction. And furthermore, the guide part of the RNA is also in the same conformation that we see in the DNA-bound structure. So it's basically sitting in an A-form helical conformation. Even though it's a single-stranded piece of RNA in this protein, it's held in that conformation, and it's ready to interact with helical DNA. And I think what we're really keen to understand now is sort of what happens at the point when a DNA molecule interacts with the PAM recognizing part of this protein, which has to somehow trigger the strand of DNA that can interact with the guide RNA to rotate around and start forming these base pairs. And I think this sort of also argues that the importance of these initial base pairing interactions is really sort of coupled to triggering that unwinding event in the DNA. So really understanding the energetics of that, I think, will be really interesting. And so for those of you so if you know some of the literature, the people doing genome-wide experiments with guide RNAs that are targeting different sites in genomes using Cas9, you might know that I think those data are very consistent with the fact that these initial base pairing interactions are very, very important for targeting. So if there are mismatches here, that tends to be very disruptive to the Cas9's ability to bind to DNA. Okay, so I want to say a little bit now about thinking about how to use structural information like this to design forms of this protein that could be regulated. And we've done a little bit of work on this. There are other really nice papers in the literature, and I wish I had gone through and more comprehensively put together lists of what people have done, but I'll try to refer you to things that I'm aware of, but I'm really going to focus on work that was done initially in our lab here. And I want to just kind of go back to the structure, because one of the things that was really clear to us in sort of looking at the crystal structures available for Cas9, as well as even these low resolution EM images, was that this is a protein that is really sort of at a gross level is comprised of two structural lobes, one of which is involved in binding to RNA over here, to the bulk of the guide RNA, and then the other lobe that contains the catalytic domains that are actually the cleavers that cut the DNA. And so I think I have, let's see. So I'm going to show you. I'll show you in a moment. But one of the other things that's interesting about the structure is that, and I'll show you this again. I showed you this on a cartoon yesterday. Is that the individual parts of these lobes that make up the structure are not contiguous in the primary sequence of the protein. So there's an interesting topology to this protein. And so in looking at the structure, we thought that maybe it would be possible to actually design polypeptides that would represent each of these separate lobes of the structure, and to then allow them to reassemble in the presence of RNA or perhaps some other trigger that would allow you to have two separate parts of the protein that form the structures that they need to form when they're in the intact state, a functional form of the protein, but where you can control when they actually come together and form that functional fold. So why do this? And so this is just outlining sort of our thinking about this. Trying to, from a mechanistic perspective, kind of back to this question about energetics, one idea that we had was, well, if it really is kind of a spring-loaded protein, maybe if you break the spring, then it doesn't, maybe it really won't have the ability to unwind DNA, but maybe it becomes a protein that can cleave only single-stranded nucleic acids or something like that. So that was one sort of thought early on about this. And then, really thinking about how does the guide RNA interact with the protein? Can we really understand which parts of this protein are most critical for binding to the guide? And then in terms of applications, of course, there's been lots of discussion about how you might deliver this protein. We talked a little bit about this yesterday. For delivery in various kinds of viral vectors, the protein is a bit on the big side, so if you could have a way of packaging smaller pieces of it, that might be useful. Also thinking about using conditional promoters so that you could turn it on only in particular tissues or at particular times where you might like to induce its activity in cells, and then also thinking about using small molecules that could induce activation. And so for all of these kinds of applications, we thought, wouldn't it be interesting if you could actually assemble this from smaller bits that could come together to form a functional complex? So this is I just wanted to remind you. So I showed you a cartoon kind of like this yesterday, but this is just illustrating the way that the individual components, the domains of this protein, are laid out in the primary sequence. And what's most striking is that the three parts of the blue domain that are one of the catalytic domains of the protein, called RUV-C, are actually quite far apart in primary sequence space, right? So that they actually fold up, they come together in three-dimensional space to form one of the active sites of the enzyme. The other active site is here, so it's in between two of these parts of the RUV-C domain, the H and H domain. And then we've got this very large alpha helical or REC domain in the center that is responsible for binding to much of the guide RNA. And so the strategy that, and this was actually done by 3 people in the lab, Sam Sternberg, Brian, who was a rotation student in the lab, and Matthias, who was a summer student, got together and said, well, maybe we can actually just use the crystal structures to redesign the way that these domains are strung together in primary sequence space. So making a piece of sort of a polypeptide that would encompass the alpha helical lobe of the protein was pretty easy, because that actually is contiguous in the primary sequence of the protein here. But designing the rest of the protein so that it would assemble correctly took a little more work. So the idea here was to take this portion of the primary sequence of Cas9 and then connect it by a linker to the very N-terminal end of the protein that provides the rest of the sequence necessary to form the REV-C catalytic domain. And so there was a fair amount of fooling around with this linker and how long should it be and what should its composition be to eventually find a sequence that would allow expression and stable folding of the protein. And this is just showing you a little bit of that protein biochemistry. So we found that we could actually express these proteins, and they purify pretty nicely. These are showing you profiles off of a size exclusion column, so they look pretty well behaved, and you could show that they can be cleaned up and purified and run on a protein gel here, and you can see that the proteins look pretty stable and clean, at least in that biochemical sense. And then, of course, the question is, are they functional? Do they work? And so this was an experiment done initially by another student, Addison Wright. And so what Addison did was to test the function of these individual polypeptides that can form either half of the Cas9 protein or the two together. And so this is and you saw a lot of data like this yesterday, where we just take a radio-labeled double-stranded DNA target sequence that has a 20 base pair target site next to a PAM, and we're just monitoring cleavage of that DNA. We radio-label the DNA at both ends so we can see the both products after cutting. And this is a denaturing gel system here to separate the substrate molecules from the products of the cleavage reaction. And these are little time course reactions incubating with either the wild-type Cas9, you get very nice cutting that I showed you yesterday. If we incubate with either of these purified lobes of Cas9 on their own with guide RNA and DNA, we get no reaction. But if we add the two together, we see a nice reconstitution of cutting. So this sort of answered one of the questions, would we have broken this thing permanently? Would it be unable to unwind DNA? It looks like that's not the case. Okay, so it can still bind and cleave a double-stranded DNA target. It's a bit slower, so this is just showing some kinetics for this. So we were looking at both using the single guide form of the RNA as well as the dual guide, so the two separate RNAs that come together to form the guide. And these are kinetics of those cleavage reactions, showing time courses for cutting. And again, what we see is that without with the lobe, one of the lobes alone, nuclease lobe alone, we really see no reaction with DNA. We only see cutting when we have both of these polypeptides present in the reaction. I just realized I really need to put my glasses on so I can actually read the slides. This would probably be a good idea. Okay. And this is the same kind of experiment using the dual guide form of the RNA. And then importantly, we found that, again, we have to have sort of the same properties that are required for the intact Cas9 to interact with DNA and cut it are true here as well. So if we have a mismatched target site that doesn't match the guide RNA, we don't see cleavage of the substrate. And if we mutate the PAM sequence in the target DNA, we also don't see cutting here. So it really looks like this is a split system that, when reassembled, really has the same properties as we see in the intact protein. And then this was an experiment that was just sort of meant to test what parts of this RNA, the guide RNA, are interacting with different, with the two different lobes of the protein. And so if you, these are binding curves. So the way we do these experiments is we're actually using just sort of an old-fashioned filter binding system where we can look at assembly of nucleic acid with these different Cas9 lobes, and we're plotting the fraction bound over here as a function of concentration of Cas9 polypeptide. And so what you can see is that when we use the full-length form of the guide RNA, so we've got the guide strand here and then the rest of the RNA that's important for protein binding, we get binding curves that look like this. So of course the wild-type protein binds more tightly, but we see some binding to this RNA by both of the individual lobes of Cas9 as well as the reassembled complex. But interestingly, it looks like the contributions of RNA to that binding interaction are different. So if we use just the, what we call the sort of the, I don't know what we call that, but it's the sort of the 3' end of the guide RNA that lacks the targeting sequence, the target recognition sequence of the RNA, just this 3' end piece of the RNA, that actually interacts nicely with the wild type and the nuclease lobe of the protein, but really doesn't bind much at all to the alpha helical lobe. And we see the opposite result when we use the other end of the RNA as the binding substrate in the reaction. So it really looks like both, the two ends of the RNA are contributing differently to interactions with these lobes to pull the whole, sort of assemble the whole complex into a functional form. Okay, and then, I don't want to drag you through this in too much, too gory detail, but really the point here is just to indicate that as we truncate the 3' end of this single guide form of the RNA, we can show that we can actually inhibit the activity of the split Cas9, the two lobes that have to come together to form an active complex, without affecting the function of the full-length enzyme. And so the point of this experiment was really just to show that, this is a destabilized, activated but sort of destabilized complex. If we use truncated forms of the RNA, we can selectively inhibit the activity of the split Cas9 without affecting function of the full-length protein. And the point here was really just to try to get to a form of this reconstituted complex that would be sensitive, perhaps, to interaction with a small molecule or something else that we could use to then stabilize a formation of an active structure. And so the idea would be potentially to take something like this, where we know that there's nothing fundamentally wrong with this RNA. It's just sort of an unstable complex here, and provide something else that would allow it to assemble stably and be active in a cell. And this is just showing that you could actually engineer these separated lobes for genome editing in cells, and this was done in this experiment just by putting a nuclear localization signal onto the one of the lobes, or actually I guess we did on both in this case, and then providing this sample, in this case, in the form of a pre-assembled RNA protein complex that could be delivered into cells. You could also imagine doing it on a vector, but we did it in this case in a pre-assembled RNA protein complex form, and you can see that there's some editing that goes on in the cells. It's very inefficient, probably because of the instability of the complex, but nonetheless, we know that this is actually active in cells. And so, there's been interest in this in other labs. So Feng Zhang's lab did a similar kind of experiment. They actually separated the protein differently. So they actually, rather than separating it into these structural lobes, they actually just whacked it in half. And amazingly, it seems to reassemble. It's kind of astounding. And then more recently, there's been a nice work showing that you can actually use these light activatable domains to reassemble yet a third split form of the protein. So I think there's lots of interest in this, and I would say stay tuned. There's lots of, I think, interesting work in the pipeline in various labs to try to make this a system that's going to be really robust for regulatable genome editing. Okay, so let me turn then in the last part of my talk this morning to thinking about RNA targeting by Cas9. And so one of the things that's interesting about the CRISPR systems is that, so first of all, there are three, kind of broadly speaking, three different subtypes of CRISPR systems. And two of those, the type 2 system that includes the Cas9 enzyme, as well as the system called type 1 that involves multiple proteins that assemble into an RNA-guided targeting complex, both clearly, I think, interact with DNA, sort of double-stranded DNA targets in cells. But there's a third type of CRISPR system called type 3 that, there's emerging data that really suggests that at least in some circumstances, that system targets RNA, and it may target RNA in the form of a new transcript coming off of a DNA. It may be that there's an R loop that's really being targeted. It's kind of not quite clear yet, but it does seem like, at least in that system, there is some evidence for RNA binding and cutting. But interestingly, in nature, the type 1 and type 2 systems do not seem to interact with RNA. Why that is, we don't really know. But I want to share with you some sort of a line of work that took place in our lab that really was the result of biochemical experiments that we were doing to understand DNA targeting by Cas9 that led to thinking about how we might actually rejigger the system to interact and cleave RNA targets. And this was I'm going to show you first just a very simple experiment that was done by Sam Sternberg in the lab. And so what Sam was doing was, these sorts of now kind of standard experiments where we take a double-stranded DNA target substrate and show that we can get targeted cleavage by Cas9. And this is a time course reaction showing cutting. We have one DNA strand labeled in this experiment. So you see one product band on this denaturing gel system. And what was interesting was that if you take exactly the same DNA strand corresponding to the bottom strand of this substrate, so it can base pair with the guide RNA, it's a very poor substrate for cutting by Cas9. And that was controlling for binding, so it still can bind and base pair with the complex, but it's not a good substrate for cutting. And so the question was, why is that? And so what Sam did was to start adding back parts of this top strand to understand what it was that would trigger this to be a good substrate. And we initially thought maybe it was really the process of unwinding itself that was somehow coupled to DNA cleavage. And it turns out that actually, as soon as Sam added back a DNA oligo that included the PAM nucleotides, the GG on the upper DNA strand here, this single-stranded substrate now becomes a pretty robust substrate for cleavage. And this is over here is just showing you that plotted in the blue bars are the KDs, sort of the binding constants for these 4 different substrates. So they all bind very similarly to Cas9, but only the 2 on the ends that include the PAM nucleotides are robust substrates for cutting. And so that got us thinking that maybe we could, and this was actually before crystal structures of Cas9 showing the PAM binding cleft were available, but we imagined that there might be some kind of a cleft in the protein that would interact with the PAM nucleotides and might in some way trigger the reaction. And so that led to thinking that maybe we could actually trick the enzyme into cutting single-stranded nucleic acid substrates by simply providing the PAM nucleotides in the form of a short oligo. And we started calling these PAMers in the lab. And so this is a cartoon showing an experiment with a single-stranded DNA target. So this is the experiment I showed you on the previous slide. But we thought, well, maybe we could also do this with single-stranded RNA targets, which are not natural substrates for Cas9, but maybe they could be if they were incubated together with PAMers that could somehow activate the enzyme. And so the thinking was that in the presence of a single-stranded substrate that could bind by base pairing to the guide RNA, the PAMer would come in and interact with the binding site for this GG dinucleotide in the protein and somehow trigger cleavage activity. And you'll note that to ensure that the PAMmer would actually be localized in the protein correctly, we also wanted to include additional nucleotides here on the end of the PAMmer that could base pair upstream of the actual cleavage site in a target nucleic acid strand. And we did a lot of sort of playing around with this and, of course, hoped that we could actually trigger the reaction by just throwing in a GG dinucleotide, but the binding affinity, I think, is just low enough that that doesn't really work. So this is, I'll just show you a few of these experiments, and I want to mention that this was done by a great sort of team in the lab. Mitch O'Connell, a postdoc, Ben Oakes, who's here in the back, was a rotation student at the time doing some of these experiments, and Sam Sternberg, the three of them kind of teaming up to think about how to do this and how to test these ideas. And I'll show you just a few of the experiments. So this is showing a comparison of some different types of substrates that are cartooned over here and their behavior as substrates for cleavage by Cas9. So of course, a double-stranded DNA is a very robust substrate, as I showed you before. If we incubate a single-stranded RNA target with the complex, it's a very lousy substrate, but as soon as we include one of these PAMmer oligos that contains the GG dinucleotide of the PAM, we can see pretty robust cutting of the RNA. And interestingly, a double-stranded RNA substrate is not cleaved in these reactions. So we really think the PAM nucleotides have to be DNA, or it could also be the case that that an RNA hybrid is just too stable a structure to really bind to this complex. The helicase is not able to open an RNA duplex the way it can interact with DNA duplex. This is a programmable reaction. So if we program Cas9 with different guide RNAs that recognize different sequences, we can show that only the RNA target that contains complementary sequence to the guide is cleaved in the reaction. So it sort of maintains this programmable property, and this is just showing that only the RNA strand with complementarity actually binds in the complex. And right, okay, so then the question was really, how do we think about using this? If we want to use this as a technology, what kinds of applications would be most interesting? And I guess in our lab, we sort of felt like the most interesting applications, at least initially, would be binding to RNA, and sort of the idea of tagless detection of endogenous transcripts. So you may know that we have, of course, we and everyone else has had great access to RNA interference, RNAi, for RNA targeting. So there's already a pretty robust method for targeting RNA and cutting it. But for binding RNA and interacting with endogenous transcripts, this has been a big challenge, and there's been a lot of effort in many labs to do this. There's now sort of a collection of methods that basically all involve using either biotinylated or tagged forms of oligonucleotides that can base pair with endogenous transcripts, and those methods can work fairly well. You can probably, maybe you've heard of CHIRP and CHART and RAT, and there's a whole series of, Luke is nodding, a whole series of these acronyms where people have been able to use sort of oligonucleotide type strategies for pulling down transcripts and then coupling that to a high throughput sequencing assay to sequence the bound fragments. But one of the sort of big questions I would say right now in sort of RNA biology is how to identify the binding partners of specific transcripts in cells, whether they're proteins or other nucleic acids, to figure out if these are part of larger complexes. And so, wouldn't it be cool if you could actually have a programmable way to interact and identify transcripts that would not necessarily disrupt native interactions they're making with other cellular partners. And so that was sort of the thinking behind this strategy here. And so the idea was to create a tagged form of Cas9 that contains a biotin, so we can easily isolate it later by affinity purification. And then we use the deactivated form of Cas9, so it has the inactivating mutations in the catalytic domain, so it won't actually cut nucleic acid. And then the trick was to provide an appropriate form of this PAMmer to trigger binding of this complex to single-stranded target sequences in RNA. And we hoped that we could actually do this in endogenous transcripts that might have, they might have varying degrees of secondary or higher order structure. They might have proteins bound at various places, but nonetheless, by identifying sites that were accessible to guides and perhaps taking advantage of some of the helicase activity of Cas9, we might be able to get this kind of a complex to form that could then be isolated and where the individual transcripts could be identified and perhaps ultimately used to find protein and other binding partners of these transcripts. And so I'll just show you one experiment that was done initially in not in live cells, but in both in a sample of purified total RNA from cells as well as in a lysate from HeLa cells to test this idea. So on the top panel, let me start here. This is basically using as our target sequence the GAPDH messenger RNA. It's a 1.5 kb, very abundant messenger RNA that's present in cells. And this diagram shows you the exon sequences in the RNA. So we could actually design guide RNAs that would target different sites within these exons. And then the experiment was to use the strategy I illustrated on the last slide to actually isolate these transcripts. And we could try it. We tried it with 4 different versions of guide RNAs targeting these different places along the transcript and tested it with and without including a PAMmer oligonucleotide. And this is actually just I think it's just a blot that shows the amount of RNA that was pulled down in each experiment. And you can see that particularly with these 2 guide RNAs, number 1 and number 2, we see a pretty nice differential between adding or not including the PAMmer oligonucleotide. And we get fairly robust recovery of this transcript with virtually no isolation of sort of background isolation of a different messenger RNA for beta-actin. So it looked like this was really a technique that could work, at least in the context of total RNA. And then to sort of ramp it up a level, we said, well, can we actually do this in just cellular lysate that would have all the proteins and other things around? And this showed us initially that when we used initial versions of the PAMmer, although we could get recovery of the transcript, we also got cleavage. And this turned out to be due to, not to anything that Cas9 was doing, but actually due to RNase H activity, which will cut an RNA-DNA hybrid and is present in these lysates. So this led to a series of experiments to identify chemically modified forms of the PAMmer that would prevent RNase H activity without blocking Cas9 binding. And by using 2'-O-methyl containing PAMmer oligos, we could actually achieve pretty nice binding and recovery of these transcripts, but without triggering RNase H cleavage. And again, very low sort of levels of background here. And so what we've been doing, so this work was published last fall, and what we've been doing since then is really to try to make the system robust enough that we can use it in either living cells or use it for things like imaging of transcripts in cells, analogous to what I showed you yesterday, but imaging RNA rather than DNA. And again, we're finding that the challenge really is ensuring that we're using appropriate PAMmers that form these stable complexes without triggering RNase H activity. We have a collaboration with a lab at UC San Diego that's had looks like some pretty nice success so far imaging RNA in cells using this kind of strategy. So, you know, I would say stay tuned. I think there's, you know, we're also doing a lot of things like, you know, doing high throughput sequencing of these samples to test really how specific is the recovery of RNA that you get in these reactions, and it looks like it's, you know, it's really pretty robust, but I don't have the data to show that to you right now.